Here is a great deer, and here is a carcass of a great deer. Well, look at that, it's a queen scavenger hunt. There seems to be a male waiting on the carcass. After mating, the male dies, and the female tears its wings off. The female then enters the carcass. The ant looks for a place full of fatty tissue. The ant then starts making a place to lay eggs by eating some of the fatty tissue. It then lays eggs. It nourishes hatched larvae and lays more eggs. The larvae which grow up into adults take roles and start helping the queen. Slowly, a colony grows. And so does an army. That is a detritus fly. It feeds on decaying matter. As it enters, it begins feeding, but it has alerted the scavenger ants, which unlike other ants, don't normally bite. Instead, they release hydrogen cyanide. Hydrogen cyanide, being more dangerous, kills any organism which inhales it for a period of time. Let's see the process and how they do this. The poison gland seems to be smaller compared to other species. This is because the ants do not sting or bite often, and instead they became almost completely dependent on releasing hydrogen cyanide. This made the poison gland almost useless. The Doofer's gland replaced the poison gland and has an extension which is where hydrogen cyanide is prepared and stored. Let's take a closer look and remove the poison gland. The extension is divided into 3 lobes, each having their own reaction. First, we will take a look at the first lobe. The lobe contains an amino acid called glycine. The lobe also contains oxygen. Oxygen and glycine react with each other. Their reaction being catalyzed by an enzyme called sythase. The reaction is as follows. After the reaction, the product produced is aminoacetic acid. HCN synthase catalyzes another reaction between aminoacetic acid and oxygen. This time the product is water, carbon dioxide and hydrogen cyanide. Here is the reaction. Cyanide is kept enclosed in the enzyme to prevent it from affecting the cells. Water is absorbed by the cells, carbon dioxide is released out through the stinger, and hydrogen cyanide is taken to the second lobe. Here an aldehyde is present, that being ethanol. HCN synthase releases the hydrogen cyanide to react with ethanol. HCN synthase then goes back to the first lobe. An enzyme called hydroxynitrile lyase catalyzes the reaction between hydrogen cyanide and ethanol, which is as follows. After the reaction, the product produced is lactonitrile. Lactonitrile is unstable, so it is quickly moved to the next lobe for the final reaction before storage. In this lobe, a nucleotide is present, that being uridine diphosphate glucose. Both substances react. This is catalyzed by the enzyme uridine diphosphate glucotransferase. The reaction is as follows. The resulting final products are linamarin and uridine diphosphate, both which are stored. For releasing hydrogen cyanide, first linamarin and uridine diphosphate are released. An enzyme catalyzes the reaction between them, that being beta-glycosidase. The reaction is as follows. The products formed are lactonitrile and uridine diphosphate. Uridine diphosphate is absorbed. As mentioned before, lactonitrile is unstable, so it decomposes into hydrogen cyanide and ethanol. Hydrogen cyanide is released through the stinger and ethanol goes back to the second lobe. For a quick explanation, in the first lobe, hydrogen cyanide is prepared from glycine by oxidation and water being a byproduct. In the second lobe, hydrogen cyanide and ethanol react to form lactonitrile. In the third lobe, lactonitrile reacts with uridine diphosphate glucose to form uridine diphosphate and linimonin, both which are stored. When cyanide is to be released, uridine diphosphate and linimonin react to form uridine diphosphate glucose and lactonitrile. Lactonitrile, being unstable, decomposes into hydrogen cyanide and ethanol. Hydrogen cyanide is released and ethanol returns to the second lobe. The cells of this extension are of two types, primary and secondary. The differences are: primary cells are the location of reaction, secondary cells are the location for storage. This is a gold fox. What we are witnessing is the fox releasing its saliva into the deer's carcass. It seems to be feeding, but by licking. The ants have begun releasing hydrogen cyanide, but what took them so long? Maybe this perspective might help. This is the fox's saliva. The ants don't seem to be releasing cyanide, so the saliva did something to the ants. The ants are now releasing cyanide, so maybe the saliva is not affecting the ants themselves. At least not chemically, but maybe the saliva did something to the cyanide release. But how can a fox release so much saliva? Well, that's because the salivary glands of gold foxes are enlarged, and also because they are naturally found near water bodies, which will be shown in the end of the video along with every other creature mentioned in this video. So now, how does the saliva stop cyanide? The saliva of a gold fox is rich in vitamin B12A, and when vitamin B12A comes in contact with cyanide, they react to form cyanocobalamin, which the fox can digest to obtain vitamin B12A. As time goes by, all that will be left is bones. When this occurs, the queen reaches the end of its time and dies. The remaining workers and soldiers will feed on what's left, that being bones, and develop into queens and males. This is possible because both male and female scavenger ants take roles as workers and soldiers, males doing the additional work of mating, but only when food supply becomes low, that is, the carcass is almost consumed. After development is finished, the queens and males leave in search of other carcasses. This cycle on average takes a year to complete. This is one of SUPROSAN's false bone-crushing dogs, specifically the brown-backed false bone-crushing dog. As the name suggests, they can crush bone. To do this, they have powerful temporals, digastric and masseter muscles. They evolved bone crushing convergently from true bone crushing Burfagnae, hence the name false bone crushing dogs. There are more than one species of them on SUPROSAN Island, and some, including this one, being social. The rest of the pack has arrived. They crush bones in order to access the nutrient-rich bone marrow. After they leave, the rest of the deer is decomposed by bacteria, millipedes, and etc. And what's left is buried and fossilization occurs, or that too gets decomposed. This is the map of the SUPROSAN landmass, it almost being the size of the Indian subcontinent. It is to be known that the beaches of SUPROSAN are so thin that they are not visible on a map. Its exact location is unknown. The passing and decomposing of the great deer shown in this video occurred in the deciduous forest called the scavenger forest, as almost every creature found here is a scavenger, or is an opportunistic scavenger. As mentioned before, these are the ranges of the creatures mentioned, and it is noticeable that the gold fox is found near a water body.